Assalamualaikum warahmatullahi wabarakatuh. My name is Hani Nadia Binti Munir, and today I'm going to present on the topic of drug discovery strategies for COVID-19. So, as we all know, uh, at the end of last year, 2019, a new coronavirus, which is identified as COVID-19, COVID-19 virus, is the etiological agent that is responsible for the 2019-2020 viral pneumonia outbreak that commenced in Wuhan city in China. So currently there is no targeted therapeutics, and the effective treatment options remain very limited. So in order for the researcher to rapidly discover the lead compounds for the clinical use, we have designed a strategy to find new drug possibility that is specific for this COVID-19. And we will explain the strategies further and I will explain the strategies further in my presentation. So before I start explaining on the strategy, I would like to explain briefly on this novel coronavirus. So this novel coronavirus is known as SARS-CoV-2 viral. So this viral genome encodes for 29 proteins, in which many of them could serve as the potential targets for the antiviral drugs. And one of the protein known as coronavirus main protease, or the short term is we call as COV and PRO, is one of the most attractive drug target for this virus, because they play an important role in the viral gene expression and replication through the proteolytic processing of the replicase polyprotein. So in my presentation, I will focus on using this COV and PRO as the biological target to identify the new drug leads to treat this COVID-19. So the first strategy that I plan is using structure-based drug design. So the structure-based drug design is a method in which we use the structure of the biological target to design candidate drugs that are predicted to bind with the target in a high affinity and selectivity. So firstly, we need to obtain the information on the 3D structure of the biological target, which is the COV and PRO, using several methods such as crystallography, NMR spectroscopy, or homology modeling. So how can we obtain the structure using this method? So I will explain one method, which is the crystallography. So the protein with the water will firstly freeze into the crystal, and this and they will be X-ray, and the picture of this protein will be taken. So from there we can get the structure of the target. So once we obtain the structure of the target, which is the COV and PRO, we can use this structure to design the candidate drugs that are predicted to bind to the targets in a high affinity and selectivity. So for the COV and PRO, the candidate drugs is the inhibitors for the COV and PRO lah. So we will use the computer algorithm to position or dock the compounds into selected pockets of the target structure. So as you can see here, this is the structure of the target that we have obtained. So from this, we need to design the drug that fit the pocket, because the drug that fit the pocket is like the most promising drug lah that can be used to be developed for treatment of COVID-19. So this compound will be then ranked based on their steric and electrostatic association with the target proteins at the selected position, and the most promising compound will be further evaluated for development into drug to treat COVID-19. So the next strategy that can be used to discover the drug for COVID-19 is virtual screening. So virtual screening is a method in which is a method that is used in the drug discovery to rapidly to identify the possible drug candidates. So in virtual screening, we use a high performance computing, which is also known as in silico approach, to analyze a large database of chemical compounds in order to identify the potential drug candidate. So the structure of the target, which is the COF and PRO, is used as the model to identify the lead inhibitors using through the in silico screening. So to achieve this, a database of the potential binding compounds will be docked to find the compound that can fit snugly into the substrate binding pocket. So as you can see here, the ligand is the compound that is tested. So the compound must fit into the substrate or the target, so that they can become, so that we can know whether they are a promising compound for development to be developed into drug for treatment of the COVID-19. So for virtual screening, we can use many various methods other than molecular docking, such as we can use pharmacophore analysis, similarity search, scaffold hopping, and many more. So, okay, for the virtual screening, we will design the in silico filters to eliminate compounds with undesirable properties, and we will select the most promising drug candidates. So the filters can be like the compound need to be high bioavailability. So for the compound that have low bioavailability, they will be eliminated and they are not the promising drug candidates. So next, for the next method, we can also use high throughput screening. So high throughput screening is actually a method that is used in drug discovery, in which we can rapidly identify possible drug candidate. And this method is actually a starting point for drug design. So high throughput screening of large number of compounds in automated fashion against specific target, which is the COF and PRO, to identify the hits. Hits is actually the compound that exert desired effect on the target. So the success of the screening is influenced by the number and diversity of the compound library. So usually the compound that finally become the finished drug product is different compared to the lead compound that emerged from the initial high throughput screening. Okay, next method or strategy for drug discovery is the drug repurposing. So drug repurposing involve investigation or study of the existing drug for new therapeutic purposes. So because the development of novel drugs is a time-consuming process, it is a time-consuming process, and usually we require several years of work for to get the clinical approval. So this method is actually a very effective strategy to combat the novel disease caused by the SARS-CoV-2 coronavirus. So the investigated drug have been approved for some disease, and it is safe for human use, and only the effectiveness against the COVID-19 needs to be established. However, we still need to do the clinical trials to ensure that such treatment is better than the placebo. So actually, there are few existing broad-spectrum antiviral agent BSAA that rapidly introduced into the clinical trial spanning phase 2 through phase 4. So actually this strategy make the process of the drug discovery of COVID-19 much more faster, because we don't have to start from the scratch to develop the drugs. So for example, as we know, remdesivir has the activity in preclinical studies against species of coronaviridae implicated in the SARS-CoV, SARS-CoV as well as the MERS-CoV, and they also has already been studied in randomized control trial for Ebola virus disease, and they show the antiviral effect. Thus, if this repurposed drug demonstrate the efficacy against the COVID-19, they could be manufactured profitably, and they can help ensure the affordable access to vital treatment for COVID-19. So last but not least is the phenotypic screening. This is also one of the method for drug discovery. So this is a method used to identify molecules with the ability to alter the cell's phenotype. So for the phenotypic screening, compared to the target, compared to the structure-based drug discovery, this phenotypic screening does not rely on they do not rely on the identity of the specific drug target. So for the phenotypic screening, uh, we actually identify the candidate molecules effect before we identify the specific biological target that is responsible for the phenotypic response. So the candidate molecule that acts on the biological target in its normal biological contact rather than on a purified target. Mm. So the key benefit of this approach is that they have a capacity to increase they have the capacity to capture the complex biological mechanism that are not otherwise achievable. And this approach also increase the understanding of the cell signaling networks that involve in the disease process, and they can decrease the amount of clinical failures in which they by identifying the factors that contribute to the unexpected activity, toxicity, and the lack of efficacy. So that's several strategies that can be used for discovery of drug for COVID-19. So that's all from me. Thank you.